So I would like to welcome everybody to our Friday morning team research forum. And I'm delighted that this morning it's going to be focused on a program that's making precision medicine for cancer more accessible to marginalized communities. I'm absolutely delighted that our speakers, Professor Luis Carvajal Carmona and Nicole Halmai, accepted to talk today. So Luis is Associate Vice Chancellor of the Office of Diversity, Equity and Inclusion, and is also professor and Auburn Community Cancer Endowed Chair in Basic Science in the School of Medicine. He is also the director of the Center for Advancing Cancer Health Equity and co-director of the Community Engagement Program at the School of Medicine. Luis is the holder of the Auburn Community Cancer Endowed Chair, and his interest is focused on cancer genetics, population genetics, genetic demography of minority groups, and functional genomics. Before joining UC Davis, he was a senior research fellow at the Wellcome Trust Center of Human Genetics in the University of Oxford in England. And he did his postdoctoral fellowship at the London Research Institute and his PhD at the University of College London. Nicole Halmai is a postdoctoral scholar in the Carvajal Carmona lab. She leads the functional genomics team, which focuses on the characterization of therapeutically relevant cancer driver variants in patient-derived models, with a specific focus on racial and ethnic minority cancer patients to address cancer health disparities. Her work also includes the use of CRISPR-Cas9 genome editing technology and patient-derived 2D and 3D cell culture models. And she's a native of Phoenix, Arizona, and graduated from Denison University with a BS in molecular and cellular biology. Without any further ado, I'll give it over to you, Luis. I also would like to ask you to please post your questions in the chat, and we will have some time at the end of the two presentations for a discussion, and we will try to address all of your questions. Thank you.
Thank you, Angela. Can you all see my screen? Yeah, okay. All right, so thank you for inviting me to present today. I'm happy to be here with one of our great talents in the lab, Dr. Nicole Halmai, who is going to be presenting more on the kind of science and precision medicine approaches that we have in the lab. In my talk, I'm going to give you an overview on why we think it's important to focus all research efforts in the community. So there are two aspects here I'm going to discuss. Well, I'm going to be speaking about cancer health disparities first. I'm going to give you an overview of what disparities are and what are the main disparities. And then I'm going to kind of present a concept that we have been developing in the lab on precision health equity, which is to merge the fields of disparities and precision medicine together for the benefit of marginalized communities. So let's start with cancer health disparities. So we talk about cancer health disparities, and typically these are differences in cancer incidence or outcomes between a reference populations and other groups. And in the case of the US, the reference population is normally used is non-Hispanic whites or the white population. And the disparities normally are associated with communities of color. So this is something that we are interested in because the communities of color in the country, their numbers are growing. So even though the US is not a minority majority state, California is a minority majority state. 62% of Californians belong to communities of color. And as you can see here, some of those communities are growing a lot, including Latinos. At the moment, the largest population group in California are Latinos. They became the largest group in 2017, they overtaking the white population, and they are expected to be around 50% of the population in few decades. So we have a particular interest in working with Latinos, addressing their needs in terms of cancer health. So this is a map of the catchment area of the UC Davis Cancer Center. We are a comprehensive cancer center, which is the highest designation of the National Cancer Institute. And one of the requirements from the NCI to us is to define our area of influence or impact. We call this the catchment area. And this catchment area is a 19-county region around Sacramento, which include the Sacramento Valley, the San Joaquin Valley, and the Sierra. So we serve multiple communities in the region. And here I'm showing you that even though this is California, it's quite a diverse population. The people living in the valleys, they are predominantly communities of color. So here in Merced, for instance, which is a county south of Sacramento, 71% of the people in Merced County are people of color, and actually most of them are Latinos. So this region, this kind of dark blue, mostly include people of Latino ancestry, as well as other minority populations like Asian American, Pacific Islander Americans, as well as African Americans, some American Indians or Native Americans. So it's quite a diverse region. The Sierra, on the other hand, it has a predominantly white population. So the cancer needs of the San Joaquin and Sacramento Valley are slightly different to the cancer needs of the Sierra Valley, and we want to serve all of them in the cancer center. Here on the right, we have another important element of this catchment area, and it's the average age of people in these regions. And as you can see, the people in the regions who tend to be more minority, from more minorities, they also tend to be younger. While the people living in the Sierra, they tend to be older. So this is important because if we are going to say a lot of our cancer patients are going to come from this part because they're older, cancer is more common in older people. So we need to serve them by say incrementing participation in clinical trials. They can benefit from our center. On the other hand, these ones, they are prime because they are young, they are prime for prevention. So we need to be proactive in developing cancer prevention and early detection programs. So we prevent cancer, or if they have cancer, we catch it early, so it's more curable. So around 5 years ago, with funds from the Office of the Provost and from the Cancer Center, I launched this initiative called Latinos United for Cancer Health Advancement because until that point, we were not doing a lot of activities with the Latino community, even though they represented one of the largest constituents in our catchment area. So we launched this with a philosophy of serving their needs and of communicating with them in a way that linguistically and culturally they will understand. Lucha was launched about 5 years ago. Now it's also co-directed by Dr. Laura Fejerman. And this has been an initiative that has been successful. It's becoming a model in the nation on how we can use the power of a comprehensive cancer center to start addressing and promoting cancer health in marginalized communities. So we do this using a number of approaches. So here, Angelica Perez, she's one of the initial founders of Lucha with me. She's a cancer health educator now working in our group in Rosetta Studies. And Alexa Morales, who is now a medical student in Davis. So she was part of Lucha, and we used to go. So Angelica and Alexa here are, native from the region. They are from the region. They speak Spanish. They are, bicultural and bilingual Latinas. So when we go there, we don't have these issues of linguistic isolation. We can provide information in a way that people, understand it and can use it. So we have a number of activities. We have, we have activities in churches, with community agencies, with family resource center, with the Mexican consulate, with health clinics, etc. And we provide information in Spanish on cancer prevention. And we basically try to, establish partnership, and we have been doing this for 5 years. And the Lucha partnership have been key for us to be effective in our mission as a cancer center, but also to actually develop larger studies like the White study. We are not going to speak about that today in detail, but this was a project that we secured because we have these amazing community links. So this is another example of what we do. We have a Genentech, a pharmaceutical company in the Bay Area. They have been kind enough to supporting us for cancer education health fairs. So we go to the community. This is an inflatable colon. I'm going to show you some data on the issues of colon cancer in Latinos. But what we do is we bring this colon. We have a comprehensive health fair covering all aspects of healthcare. And so we go there. We bring our bilingual and bicultural staff. In many cases, also we bring UCAB doctors that can answer questions or medical students, and we provide interventions aimed at increasing colon cancer awareness, colon cancer education, and now in the next phase, we are also going to provide the participants who are eligible and not up to date with colon cancer with kits that they can go to their local clinic and be tested. So this is actually something that we are doing actively and is contributing to removing myths and issues that people have for accessing cancer prevention, and I think that has been quite successful. Something very exciting that we are also launching soon, or which is about to launch, is a collaboration with Woodland Community College and with an NGO called the Health Education Council. In collaboration with us, we are launching a program to train promotoras or community health workers on cancer health equity. We have a 750,000 grant from Gilead, another pharmaceutical company, to establish this program. So the program is going to be taught by UC Davis faculty. We are hiring a course coordinator, and this is going to be free and accessible to promotoras throughout the regions. And we are going to tell them or train them in multiple topics that are important for health promotion, for health equity, and for in the case of cancer, for everything about cancer education. So the certificate, we are hoping to launch it later in the year. And they are going to take the certificate, the promotoras, and then these promotoras will become ambassadors or embajadoras of the new center we launched at the school, which is the Center for Advanced Cancer Health Equity. And these embajadoras are going to be linked to communities or to local clinics, and they are going to help us link activities in the cancer center with community activities. They are going to help us establish patient support groups, and also they are going to help us establish more education and ideally in the future, increasing local participation of these patients from marginalized communities in clinical trials in the cancer center. So it is going to be an incredible program. We have two great community and education partners. And I think also this is going to become a model, because what we learned from the pandemic is that most of the healthcare happens in the community. And if we want to be effective as a health system and an academic institution, we need to leave the ivory tower of Davis and go to the community and provide them with opportunities so they benefit from all these great things that are being done in Davis. So let's now talk about cancer health disparities. This is something very important for Davis, for our cancer center, for the new center, and our differences, as I said, in outcomes between mostly people of color and the reference population. But not only is about race or ethnicity. I can tell you here, for example, showing that Black men tend to have twice the risk of cancer, prostate cancer death when they compared to white men. But also, for instance, people living in rural populations who are mostly white, they also have disparities. And a case here is, men living in Kentucky, the incidence of lung cancer is 3.5 higher than people living in urban areas. So disparities affect nearly every group in this country. They are more common in people of color, but also there are other components driving disparities. In this case, in our region, we see disparities in the Sierra, mostly on access to healthcare, and we also say disparities in the San Joaquin and Sacramento valleys. So these are complex multifactorial phenomena driven by multiple factors. So there is the environment. So in our region, the environment is wildfires, air pollution, issues with water contamination. Those are elements that increase cancer incidence and mortality in some regions. Behavioral, mostly lifestyle and tobacco, those also lead to disparities. For instance, we have higher smoking rates in African Americans that may partially explain why they have more cancer. In Latinos, we have lower smoking rates. They tend to have lower cancer rates in general. social factors, education, income, and employment that determines if you can actually have insurance, if you can go to your doctors, if you are well taken care of, you have good quality health care. Cultural factors, in Lucha, we are working a lot about them because at least in the Latino community, cancer is a word that we don't like to talk about. So we are trying to identify the barriers and overcome them so that people actually can be up to date with their screenings. And also biology. So Dr. Nicole Halmay is going to talk about those biological drivers of disparities, but we are also working hard on that here in Davis. So this is for Latinos, these are the cancer incidence rates in the top and mortality rates in the bottom for men on the left and for women on the right. And as you can see, the most common cancers, well, we call them the big four because they account for around half of all the cancer patients, are breast, prostate, colon, and lung cancers. So those are the big four in terms of incidence and mortality, and also they are the big four in terms of incidence and mortality in Latinos. If you can see, prostate, colon, and lung rank very prominently in Latino men, as well as breast and the other ones in Latino women, right? And they also are leading cause of mortality. However, when we look at the same rank of incidence and mortality in white, which is showing here with these numbers in blue and in red, we see some differences, right? So the most obvious difference, for instance, in Latino men, is that liver cancer is the 13th most common cause of cancer incidence in white men, but it's the 5th in Latinos, right? Same in terms of mortality. Is the second cause of mortality in Latinos, liver cancer, the 5th in men, and stomach cancer, it is the 5th or 6th cause of mortality in Latinos, but it's the 14th in white men. So even though cancer affects all the populations, there are particular cancer types for which we see bigger differences, and that's what we call disparities. So this is a table showing you the disparity ratios for men and women in terms of incidence top and mortality bottom. And let's take, for example, incidence. For liver cancer, Latinos are 71% more likely to be diagnosed with liver cancer and 47% more likely to be diagnosed with stomach cancer, right? In terms of mortality, Latino men are 47% more likely to die of stomach cancer, liver cancer, and 103% more likely to die of stomach cancer, right? So the cancer burden is higher in Latinos for these two cancer types. Same for women, and I would say one of the largest disparities is the mortality for stomach cancer that we see in Latinos, okay, which is around 2.5-fold higher or 150% more, right? So these are important disparities, and we, as a comprehensive cancer center, have an obligation, responsibility to do research that addresses these issues. This is data from our catchment area, the 19 counties that I show you. And as you can see, stomach and liver are very prominent. So the data that I show you here is not Latinos nationally. This is the data from local Latinos. We are also looking at data from the California Cancer Registry. It's a paper that we published around 2 or 3 years ago, where we look at the characteristics of cancer patients in the state who were whites and Latinos, and we saw big differences. So even though it's called stomach cancer or gastric cancer, the epidemiology and the characteristics are different. For Latinos, we see a lot more women patients with stomach cancer. We see a lot more young patients with stomach cancer, people diagnosed under age 50. They tend to be from low socioeconomic status or from marginalized communities. They tend to also have more aggressive manifestations like diffuse histology is a bad histology to have, as well as having metastatic tumors. So there are big differences in the presentations of this cancer, which is a disparity associated cancer affecting the communities that we are meant to be serving here in Davis. So we also did something interesting in Lucha. Before the pandemic, we interviewed around 350 Latinos, and we were collecting information on health information and outcomes. This is a paper I invite you to read. It was published in Frontiers in Genetics last year. And a simple comparison of comparing US-born Latinos and foreign-born Latinos, we saw big differences in everything, right? So this is something that also helped us further refine the community outreach and engagement approaches that we should be doing in the center, and also refocusing on research. So, we did a similar survey in the Cancer Center by, instead of going and meeting them where they live or where they are, the Cancer Center sent 10,000 surveys to randomly selected homes in the region. Those 10,000 surveys, we only have an 8% response in that survey. In this particular study, we have a 95% response. They will look at me or they will look at Angelica. We will speak to them in English or in Spanish, what they prefer, and they were always happy to answer these questions. So this was an effective way to obtain data, primary data from our communities. In the survey that I was mentioning, that we sent the 10,000 surveys, the 10,000 households, only 8% responded, and a small fraction of them were Latinos. And most of the Latinos that responded were English-speaking Latinos, US-born Latinos, who were quite different to this population that we were sampling. Let's see what we have here. And these are the characteristics. So we have, the immigrant Latinos, they would tend to be younger, they tend to have lower English proficiency, lower education, and lower income. So those are important social determinants of health that affect cancer outcomes, and we need to come up with programs that, that we can work with them. And also in terms of screening, that's why if you remember that colon cancer, that inflatable colon we bought it for a reason, because in the survey, we knew that the screening rates for colon were particularly low, okay? For immigrant Latinas, which are a huge community here, the screening rates for this particular group is less than 40%. Less than 4 in 10 eligible Latinas are up to the colon cancer screening. Okay, so we got this primary data from the community, and now we are doing this intervention. And in the intervention, we are looking at pre and post outcomes for education, but also we are now building other components, which is on acceptability of the testing and outcomes. And at some point, we are going to be bringing more precision medicine, like risk prediction using genetic information, etc. So we are building slowly a program around this issue, but this issue was identified by a close collaboration and interactions with our community. So this is the final couple of slides from my presentation before we move to Nicole's. So this is a concept that we are developing, and this has been a problem in the majority of academic medical centers in the country, which is we have amazing research, but this amazing research is happening in silos. And for cancer disparities, there are 2 elements that can help us advance this. One is in understanding the causes of disparities. On the other hand, is coming up with some of the solutions, including precision medicine. Precision medicine to prevent people getting cancer, or precision medicine to treat people with cancer so they can have a healthier and longer life and happier life. So this is a silo that we need to break by joining efforts of basic translational scientists in the precision medicine aspects, as well as people doing more community or population-based research in the disparities aspect. So this is something that we strive to do in the lab. So my lab has the philosophy of from base to community. So everything we do is because we have a problem outside the walls of Davis that need to be solved. So that was something that I was wanting to convey before me finishing. So as I said, our base to community approach is focused on addressing local needs using bilingual and bicultural approaches. We use secondary data like registry analysis and primary data like interviews from people to identify problems and to address, to ideally contribute for these communities to benefit from precision medicine and the great advances that we are having in Davis. Okay, so that's my final slide. And these are my group. I also wear my hat of Associate Vice Chancellor for Academic Diversity, as well as the Associate Director for the EI at the Cancer Center. So we also believe that cancer disparities and health equity can only be achieved if we train and promote the careers of people from the same communities. So we make a big emphasis in training as much a diverse group. And as you can see, our group is very diverse. And these are the the people or collaborators. We have a lot of great collaborators, but especially our community partners, they are amazing. And without them, we wouldn't be able to be successful. And of course, the people funding our studies. Thank you. So I think what I'm going to do now is I'm going to pass it to Dr. Nicole Halme, and then we will have, I think, questions at the end, Angela.
Yes, yeah, we can do that unless somebody has something really, really burning that could be asked quickly. No, all right.
No, yeah, we only have a couple 20 minutes per talk, so let's over to Nicole.
Please
Thank
post
you.
it in the chat. Thanks.
Great, thank you. Thanks for the introduction, Luis. So yeah, I'll keep it going. So my name is Nicole Halme. I am a postdoc researcher in Luis's lab. I want to thank the organizers for allowing me to speak today about our efforts to establish and characterize patient-derived models from racial and ethnic minority gastric cancer patients to advance cancer precision health equity. So Luis mentioned precision medicine, and I just want to give a very simplified overview of that idea. So through increased cancer patient sequencing and epidemiological studies, we've been able to identify risk factors that contribute to the development of cancer, but also been able to stratify cancer into molecular subtypes that are therapeutically relevant. And together, this has allowed us to develop early detection screening, as well as patient sequencing of critical mutations that together provide a clearer view at the individual level of what types of preventative measures and treatments are going to be the most effective. And gastric cancer, as Luis mentioned, is definitely one of those cancers that has benefited from the precision medicine movement. We know that there are a number of risk factors associated with gastric cancer development that range from environmental, lifestyle, genetic, and societal. And through large cohort sequencing efforts, such as that of the Cancer Genome Atlas, or TCGA, we've been able to identify 4 molecular subtypes for gastric cancer. This includes chromosomal unstable, Epstein-Barr virus positive, microsatellite unstable, and genomically stable. And each of these have a unique histological and mutation and expression landscape. And importantly, we've been able to associate these molecular subtypes with patient survival and therapeutic response. So I'm showing stratification of the molecular subtypes with overall survival here at the top, and then on the right, stratification by response to adjuvant chemotherapy. So one thing that Luis mentioned, and that's really important for our lab, is that despite all of these advances with precision medicine, especially in regards to gastric cancer remains a significant problem worldwide and also in the US. So worldwide, as of 2020, it is the 5th most common cancer type, with about 1 million cases each year, and the 4th leading cause of cancer-related deaths. And despite our advances, gastric cancer has a particularly poor prognosis. There are relatively few symptoms that patients experience in the early stages when it's more treatable. So most patients are diagnosed at advanced stages, where in the US, the 5-year survival rate for metastatic gastric cancer is just 5%. Additionally, compared to other solid tumor types, there are relatively few FDA-approved targeted therapeutics, and we actually see this as an opportunity to identify new therapeutics that might be more effective. Also, as Luis mentioned, gastric cancer is a significant health disparity for US racial and ethnic minorities. And I'm showing you SEER data here on the right for incidence. And you can see that all racial and ethnic groups compared to whites and non-Hispanics have higher incidence rates. It's the same for mortality rates. And in addition to that, as Luis talked about before, we know that the clinical presentation in these racial and ethnic minority groups is different from non-Hispanic whites, so often diagnosed at earlier age, more advanced stages. We often see higher frequencies of infection-associated gastric cancer, so Epstein-Barr virus positive and H. pylori positive. We also see higher frequencies of non-cardia and diffuse subtype. So this is all evidence really that racial and ethnic minority groups may experience a unique combination of these risk factors for gastric cancer. And the idea is that if we can identify what these differences are, we might be able to start taking steps towards reducing these disparities for these communities. Unfortunately, one of the major problems is with the tools that we have right now for precision medicine. So a lot of our large patient sequencing cohorts, such as that of TCGA, and the preclinical models that we use, such as established cancer cell lines, don't represent the diversity that we know exists in the US. And just showing an example here, I have on the left, according to the 2020 US Census, the distribution of race and ethnicity in the US population, as well as what we would expect to see in representation of gastric cancer patients based on relative incidence. And you can see for TCGA here, for example, the same distribution of race and ethnicity as well as by genetic ancestry. And then for the COSMIC database, which houses genomic data on established cell lines, that there's significant overrepresentation of non-Latino white and European ancestry samples, and relatively little representation of racial and ethnic minority community samples. And I just want to point out, so Luis mentioned that we're particularly focused on the extreme disparities among the Latino population. And despite the Latino population making up nearly 1/5 of our entire US population, in the TCGA cohort for gastric cancer, there were just 5 individuals that self-identified as Hispanic or Latino. So one idea to fix this is if we can increase the inclusivity and the diversity of the cancer patient cohorts that we use as input for our identification of risk factors and creating the molecular subtypes, we might actually be able to move towards this cancer health equity model that includes race, ethnicity as a risk factor and allow us to identify more inclusively the molecular mechanisms that might underlie why a person would respond to a specific type of therapy. And I just want to mention, I would argue that race ethnicity really is distributed through all of these other categories of risk factors: environmental, lifestyle, genetic, and societal, because as social constructs, it not only defines who our ancestors are in thinking about genetic ancestry, but also what things do we have access to, where do we live, and what type of food we eat and who we interact with. So that idea has really driven the project that I'm going to describe today in the Carvajal Cremona Lab, which is a multi-center collaboration between a number of UC comprehensive cancer centers, as well as cancer centers across the country. And the project is called the University of California Minority Patient-Derived Xenograft Development and Trial Center. And the idea is to generate 200 patient-derived xenograft models, which are where we take tumor biopsies from patients and implant those into immune-compromised mice, as well as patient-derived organoid models, where we take the tissue and culture that in vitro in 3D models for bladder, gastric, lung, and liver cancer, with at least 60% coming from racial and ethnic minorities. And the idea is to be able to take these models, characterize them extensively with molecular characterization as well as preclinical therapeutic screening in order to identify genetic drivers of cancer that might be specific to racial and ethnic minority patients. And ultimately, the goal is to contribute to the diversification of preclinical models and molecular data to include racial and ethnic minorities. And I want to mention that all of the models that we're generating in this program are being deposited with the NCI PDX-NET network to be available to other research community members. And so today I just want to talk a little bit with the remaining time of some of the preliminary data that we have from our gastric cancer patients. So thus far, we've been able to collect a cohort of 24 gastric cancer patients in this study. And here you see the stratification by race and ethnicity. This is self-identified race and ethnicity on the left for our UCAMP cohort, and then on the right compared to TCGA. And I think what you can appreciate is that already we have a lot more representation of racial and ethnic minority populations in our samples, specifically incredible representation of the Hispanic Latino community in our patient population. And so what are some of the differences that we're seeing here? Well, when we look at, when we're able to look at the mutation profile file, and with a specific platform created by a project scientist in the lab, Ted Toll, we're able to assign molecular subtypes. And so when we look at subtype distribution between our cohort and the TCGA, we see striking differences already. So while the TCGA was mostly dominated by the chromosomal unstable subtype, with relatively fewer cases of the other three major subtypes, MSI, genomically stable, and EBV positive, on the other hand, our UChem cohort seems to be dominated by the genomically stable subtype. And you might not have been able to see it earlier in my slides looking at the molecular subtypes, but the genomically stable subtype is actually the most aggressive and treatment resistant of the 4 subtypes. And I'm not showing it here, but when we look specifically at these genomically stable samples, we see a significant enrichment in Hispanic Latino patients. So we believe that this increase in this subtype is really driven by our Hispanic Latino patients. When we're creating models for these, so patient-derived organoids and PDXs, we have had success in developing, in generating models for all 4 molecular subtypes. So we have subtype stratification for patient tumors, PDXs, and organoids. And we've been able to utilize these to better understand the mechanisms that might underlie why we would see differences in certain molecular subtypes in these communities. So when we look specifically at what genes are most frequently mutated, specifically when we look at known tumor suppressor and oncogenes, we also see, not surprisingly, differences compared to TCGA. So what we see here in blue is the UCAMP samples, and in yellow is TCGA, and these are frequencies of cases with mutations in these driver genes. Interestingly, we see an enrichment in a number of Wnt signaling genes, so things like APC, RNF43. We see fewer samples with TP53 mutations, which corresponds with fewer cases that are chromosomal unstable. TP53 is the major driver of that subtype. We also see increases in tumor suppressor genes like CDH1, which again is not surprising when you look at the molecular subtype distribution, because CDH1 is one of the driver genes associated with the genomically stable subtype. So looking at these differences, we were interested to see if we step back at a pathway level, what kinds of differences we saw. And we saw a significant enrichment in 4 major pathways in our cohort: the PI3K-AKT pathway, CDK cell cycle pathway, Wnt pathway, and RAS-MAPK pathways. And importantly, there was a lot of overlap in alterations between multiple pathways. And particularly, we saw a significant overlap between the PI3K-AKT pathway and CDK cell cycle pathway. So I mentioned that we perform preclinical therapeutic screens, and this is an example of those screens. So we include a number of targeted therapeutics as well as chemotherapeutics. And what I'm showing you here are the results for 4 organoid lines that we have from minority patients that have both PI3K and CDK alterations, compared to 1 line that does not. And what we can see very nicely is that the models that were shown to have these somatic alterations do seem to respond to the correlating therapies. So PI3K inhibitors, AKT inhibitors, and CDK4/6 inhibitor. So we can take this a step further in trying to understand in an in vivo setting. So we can use PDXs to also show this same treatment response. So here is one of those lines that we were able to create a patient-derived xenograft model in mouse models. And you can see that compared to vehicle, there is a reduction in tumor growth when we treat with CDK and a CDK and AKT inhibitor. And this did not show to have any significant toxic effects. And when you look at protein expression, we're able to see corresponding downregulation of those pathways. So in thinking about what might be, better treatments for these populations, these kinds of studies are showing what treatments might help inform clinical trials for minority populations. And then taking it even a step further to really understand what are the mechanisms of resistance, we can take those lines that are sensitive to our treatment. So in this case, taking the CDK inhibitor treated line that was sensitive to that, treating those lines with a reduced amount of that therapy to allow the cells to become resistant. And so here you can see we were able to create a resistant line of this PDX model here that no longer responds to the CDK inhibition. And we are using these lines that we generated to have resistance to these specific therapies and sequencing those to understand what might be the driver mutations that are causing resistance to help us understand in the clinic what mutations we should be looking for patient response. And then at the same time, showing a little bit of corroboration between these two platforms with the help of a very talented junior specialist that Luis already introduced, Alexa Morales-Arana, who is now a pre-med or is now a medical student at UC Davis. We've been able to use the CRISPR-Cas9 genome editing system to take our organoid lines, normal gastric organoid lines, and knock out these driver genes. And so in this case, we used guide RNA targeting CDKN2A, which is a major negative regulator of the cell cycle pathway, where we can create knockouts of these genes. Here we have a 1 base insertion. And when you look at the organoids, I hope you can appreciate that compared to wild type, we see differences in morphology that might be indicative of carcinogenesis. And so we can use these two platforms, PDXs and PDOs, to do functional validation of candidate driver mutations and of resistance to better understand what might be driving these differences. And lastly, I just want to talk a little bit about how we're also looking at initiation mechanisms using our minority patient-derived organoids and functional genomics. So I mentioned that we saw a significant enrichment in alterations in the Wnt signaling pathway. So what we did here is we took normal gastric organoid lines. We knocked out both APC and TP53 in our lines. And here is an example of what those look like compared to wild type. Again, you're seeing differences in morphology, maybe indications of alterations that are looking more carcinogenic. When we look at expression data, we can see that our knockout clones have aberrant signaling of Wnt signaling through overexpression of beta-catenin, even in the presence of no Wnt elements in our media. Same thing with RNA expression. We have overexpression of downstream signaling genes in both Wnt signaling and in the p53 cell cycling. We see the same thing when we look at immunohistochemistry of these organoid lines. And so now what we're doing here is looking at initiation mechanisms, basically by taking these knockout lines. So these organoid lines that have the combinations of mutations that we see in our minority patients, keeping those in culture for a long time, in this case about a year, and then looking to see what alterations occur with snapshots over that year in culture to see what types of mutations, copy number alterations are in RNA expressions that might be indicative of carcinogenesis. So in conclusion, I've given you an overview of our UCAMP Trial Center, and really it's an infrastructure to address cancer health disparities in US racial and ethnic minorities. We're using both PDXs, our xenograft models, and PDO organoid models generated from racial and ethnic minorities with gastric cancer for molecular characterization and preclinical therapy screening. And already we see in our initial gastric cancer cohort an enrichment of a highly aggressive and treatment-resistant subtype, enrichment of alterations that seem to respond to their corresponding therapies, both in vitro and in vivo, and enrichment in key pathways that we know result in tumorigenic transformation. And so as we're moving forward, we're trying to incorporate also epigenomic and transcriptional somatic alterations. We know that it's not just mutations that can cause tumorigenic transformation, but the epigenome is also a critical component of that. We're also doing mechanistic modeling of cancer initiation and therapeutic resistance and sensitivity using our genome edited organoid and PDXs. And ultimately, our goal, as Luis mentioned, is to improve our understanding of genetic drivers and effective therapies for racial and ethnic minority cancer patients, ultimately to advance towards cancer health equity. And I just want to thank everyone in the lab, specifically my mentor, Luis, who has just been an incredible support system for me as I move through my career. Everybody in the lab who I mentioned, specifically Hong Yang, who does a lot of our PDX work, Paul and Ted, who have been our bioinformatics support, Anna, Alexa, Alma, who have all been important in the PDO and PDX generation, as well as our collaborators at other institutes and all of our collaborators here at UC Davis, particularly the UC Davis Mouse Biology Program, who does a lot of the heavy lifting in our xenograft model work, and of course the funding sources. And thank you, and happy to take any questions.
Thank you very much, Luis and Nicole. These were really excellent presentations. I forgot to mention that this session was part of a series that the School of Medicine has been organizing together with the Vice Chancellor for Research Office, and we've been showcasing our awardees from the Kruger Wyatt Settlement Award, and Luis has been one of the four principal investigators and awardee in this program, and this is part of that funded program, or that funded program is also part of Luis's larger program. Actually, I would like to start with a question that's somewhat related. I'm just curious if I know that Nicole, you focused on gastric cancer, but I was wondering if you discovered some molecular links between gastric and breast cancer that's also typical of Latino populations, especially women.
Sure, yeah, I think Luis would be able to speak to this a little more because of his collaborations with Dr. Fejerman. I can just say that I think a lot of the clinical presentations that we see in our minority population, so younger individuals with more advanced stages, is seen through a lot of different cancer types. But maybe Luis can speak to a little more about the molecular distinctions.
Yeah, so I we don't have many hard data on that project, on the Wyatt project, because we are just starting. But yeah, I mean, and also I think Laura Fejerman presented that last time. But at least the paper that I presented here, which was the survey that we carried out in Lucha, there are many cancer needs, many cancer health education needs, many cancer prevention needs. So it is in Lucha also we have a community advisory board that work with us to identify and refine priorities. But at least in Latinos or in communities of color in general, there are multiple issues, and they typically are reflected with the fact that the fraction of advanced cancers that are for preventable these conditions like breast or colon or lung or cervical is more common in them, because either we have a combination of multiple social determinants of health elements that go against that does not allow them to have a healthier life. One of them is that we have a large fraction of undocumented immigrants who unfortunately do not have they don't have access to healthcare, right? So there are a number of initiatives from the state government that are hoping, with them we will hopefully address those issues. For instance, if you are an undocumented immigrant in the new state budget, you can have medical if you are 50 or older. Okay, so no matter if you are undocumented, so that would be a big thing for us in the cancer area and in many chronic diseases areas, because 50 or just around 50 is where we can start to have interventions for prevention, right? So for colon cancer, until 2 years ago, the screening age was 50, right? Now it was lowered to 45. But for most of these conditions, we can they can have access to healthcare. But of course, there are other barriers, like linguistic barriers, right? So most of these issues that we see are driven by social determinants. There is also some biology and some genetics, but mostly social determinants. And we see the most obvious impact in the preventable cancers, where they come with advanced tumors and the children becoming with advanced tumors, you know, at least at those rates. I don't know if that answered the questions.
Yes, actually, I think you gave a very, very comprehensive answer. And I did have a question from the beginning from your presentation as well, talking about gastric cancer, because if I remember correctly, also in Japanese populations who are high socioeconomically from very high tier, there is also a high prevalence of gastric cancer. So so some of the aspects, and actually I think when you showed those pie charts, Nicole, I think it would be really interesting to bring together the molecular and the social layering of the populations, and see how they related. That could actually answer some of these interesting things that have been intriguing the medical community for many years.
Yeah, actually, we are also making models from Asian and Pacific Islanders as part of the study that Nicole has, and actually we have models in Japanese patients. So yeah, so you know, we have we mostly talk about Latinos, but our efforts are comprehensive, right? And with Japan, it's interesting because we have, you know, an older immigrant population here in California that may have a different risk profile than the population of new Japanese immigrants. So in terms of prevention for gastric cancer, you are right. There is high incidence of gastric cancer in Japan, mostly driven by a high frequency of prevalence of Helicobacter pylori. Okay, so the only two countries in the world that have national screening programs using endoscopy, open endoscopy, for gastric cancer are Japan and Korea. Okay, but at least in immigrant populations in the US, like Chinese, like Japanese, like Koreans, like Latinos, because they come from high risk, high incidence country, there has been some argument for us to consider a screening programs targeting these populations. And that would be something that I would love to do here in California, to test that, because here in this part, we have a lot of Asian immigrants, a lot of Pacific Islander immigrants, and we have a lot of Latino immigrants, all of them who could benefit from well-designed prevention strategies.
Yeah,
Nicole, I don't know if you want to add.
Yeah, and I think just to add to that, I think one of the reasons why we're really excited about adding the epigenome layer into our understanding of,
exactly what I wanted to ask.
Yeah.
Exactly. Yes.
Yeah, because we know, for example, the EBV subtype, which is an infection-associated subtype, has a very unique DNA methylation landscape. And so when we think about increased infection-associated cancer in our minority populations, I think that's really is going to give us a lot of important information. So we're doing DNA methylation analysis right now on our patient samples and our models. So hopefully we have that information soon.
Yeah, this is so exciting and fantastic. And Luis, in your slide, when you, I think it was 6 different components, and the molecular, I mean, the genetic was the last one, but cultural, the socioeconomic, and the others, nutritional, etc. I would love to see something like that translated into epigenetic changes, how all of those affect epigenetic processes that can actually insert themselves on top of everything genetic, because now everything is, we know much more about epigenetics and their seriousness. Anybody else would like to comment or question. I also see Kent Lloyd here. It's really wonderful, and I'm sure you participated a lot in the animal experiments.
Maria have a question.
Hi. Thank you, Luis and Nicole. This was really excellent, and yeah, thank you for doing this work. So important. My question was, who, in addition to you, contributes to the UCamp database, and what is the process? How much data is needed? What is the process to really get to a clinical trial state for minority patients?
Oh, okay, thank you, Maria. So the UCamp is a consortium of 6 centers. So in phase 1, we have 4 of the 5 comprehensive cancer centers in the UC system. So we have UC Irvine, LA, San Francisco, and us, and also UT Southwestern in Dallas, Texas. In the new phase, we are trying to secure the funding for that. We will also have UC San Diego. Okay, so these are groups of scientists and clinicians who are contributing tumors from minority patients mostly, and we either implant them locally or they are sent here to be implanted by Dr. Ken Lloyd's morphology program. And actually, I'd like to thank Dr. Lloyd for his great, amazing support in the program. So once they are created, there are 2 projects that are developing this kind of combinations and doing the preclinical study. That is like what Nicole showed. And we have a number of clinicians, right? So in the next phase, we have now a number of compounds that are ready to go up to at least phase 1 trial. So in phase 2 of this project, hopefully, Mariel, we are going to start doing the actual translation, which is from the preclinical studies in organoids and PDXs to clinical trials in the patients that are seen by our 6 cancer centers initially, and at some point we may go nationally, but at least these early phase clinical trials are going to hopefully be involving local patients.
So then let me do a quick follow-up to that. Because reaching the minority populations is so difficult, how will you integrate your interventions, your culturally sensitive interventions to actually convince the patients to join clinical trials?
Well, one of the things that we live from we live by example. All of us are Latinos or minorities, or most of us. So we have that sense in sensitivity and trust. But also, Mariel, that's why the program that I mentioned in the presentation about this Embajadora program with the local federally qualified health centers, they are going to be like our link, right? Our link to identify local people in the community who may benefit from this, and they will be in good sync with the cancer center. So we come up with approaches that make them feel fine and make that we will start gaining their trust, so they participate in these trials. But that is the center of us. So every trial, every study we design, we'll have a lot of consultation, and we'll have the community at its center. So ideally the clinical trials. I mean, for phase 1, we have a Sacramento-wide collaboration on clinical trials at the cancer center that has been very effective. It will depend on the stage of the trial. But I think for the phase 1, where you don't have a lot of any participants, we will need to do the footwork internally in the cancer center. If we expand them, hopefully we will be able to benefit many people around the area, many patients.
Thank you.
Thank you so much again. This was very fascinating and exciting. So if there are no more questions, we are out of time also. I would like to thank also the audience for the participation, and hoping to see you all next time. The last of the Kruger-Wyatt team presentations will be on May 12th, so don't forget to join us again. And Luis, Nicole, thank you for the wonderful presentations.